Hello everyone, and welcome to Lecture 9.5. This is a continuation of Lecture 9, which focused primarily on mouth parts and specifically teeth in vertebrates. Now, because teeth are strongly associated with the skeletal system in the vertebrates, the second half of the Lecture 9 series is going to focus on skeletal systems themselves. Now, we're going to start our discussion with an overview of why support structures are necessary in the first place. All organisms have support structures which not only provide a scaffolding for the organs and the shape of the body, but they also facilitate movement, which is one of the most important and probably least appreciated functions of support structures in animals. Okay. Now, in multicellular organisms, these support structures support and anchor the organs, right, keeping them from bouncing around inside the body. They also provide attachment points for the muscles, and this is where that connection to movement comes into play. Okay. Muscles pull against the support structures of the body to move appendages, to move the head, to move the organs around if necessary. All of this facilitates movement of the different parts of the body to achieve an animal's needs when it comes to navigating their environment or repositioning their bodies for, say, comfort. Okay? These support structures in general are called skeletons, and there are multiple different types. The three major classes of skeleton that we are going to discuss are hydrostatic skeletons. We'll use cnidarians and annelids to provide examples of this type. Next are exoskeletons. Arthropods and mollusks are most famous for this sort of skeleton. And then finally, endoskeletons, which are well known from the vertebrates. All right, let's start by examining these hydrostatic skeletons. Now, as the name implies, the hydrostatic skeleton is made up of water, hence this prefix hydro. Okay. Static means stationary or stable. In this case, water is held static inside of different chambers in the body, different body segments. In this case, these guys don't have a coelom, but they do have an empty gastrovascular cavity inside their bell. When the muscles, which are called contractile fibers here, when they're relaxed and the bell is open, water is allowed to enter it and fills it up. Then when the jellyfish wants to move, these contractile fibers, which are circular, they squeeze shut, pulling in the sides of the bell, pushing the water out back here in the direction of the tentacles, and pushing the body forwards, right? Water is pushed backwards and it pushes the body forwards. You see this kind of movement facilitated by a hydrostatic skeleton in many animals, including the nematodes, the cnidarians and annelids as seen here, and also in the squid. Remember the jet propulsion that we saw through the funnel of the squid that we dissected. All right. Here's our second class of skeleton, the exoskeleton. Now, we've talked about this a bit already in class, but when you have an exoskeleton, everything is sort of positioned opposite of what we are used to. We're vertebrates. We have an endoskeleton. Our bones are on the inside of our body, and our muscles sit on the surface, the outside of our bones. Exoskeletons are a little bit backwards from our perspective. In this case, the skeleton is on the surface of the body, and the muscles are inside of it. They attach to the inner surface of the skeleton instead of the outer surface like you see in us. Okay. So when muscles, in this case, flex and extend, they're having to pull on the inner surfaces of these pieces of exoskeleton, forcing them to either come together when they want to bend a joint, or to be pulled flat when they want to extend a joint. Now, exoskeletons, as you know, are non-living parts of the body. Okay? They don't grow when the rest of the animal grows. This is one of the reasons why arthropods who have exoskeletons are part of the ecdysozoa. It's because they perform ecdysis, molting. In order for them to grow larger, they have to grow a new, larger exoskeleton. Here's how the process works. Okay? It has 5 stages: intermolt, apolysis, secretion of molting fluid, new cuticle growth, and finally, enzyme activation and the shedding of the old exoskeleton. Here's how it works. We start with the intermolt stage. During intermolt, it's business as usual. The animal's exoskeleton fits it just fine. It's not crowded inside yet. It's able to grow and increase in size, and the exoskeleton still fits. This is like buying clothes that are a little bit larger than you, so you have room to grow into them. Okay? Now, during this stage, the epidermis and all of the tissue down here is growing in size as the animal grows. But the cuticle of the exoskeleton, remember, stays the same size. When the animal's living tissues have grown large enough and it's putting pressure on the inner surface of the exoskeleton, that's what tells the animal's body it's time to molt. Right? That triggers the first stage of ecdysis, which is called apolysis. This is the separation of the living epidermal tissue, these living cells, from the non-living inner layer of the cuticle. Okay? Space is going to open up between the epidermal cells and the cuticle, and that space is going to fill with fluid. That fluid is called molting fluid. Okay? That molting fluid is going to increase the distance between this living cell layer and this non-living cuticle layer, and it's going to give that cell layer room to start growing even faster. Okay? As this fluid is secreted and it pushes the old exoskeleton up and away from the epidermis goes into growth overdrive, meaning the cells start dividing really, really quickly, creating a whole lot of new epidermis. Now, this new epidermis wrinkles because there's so much of it, there isn't enough room on the body for it to lay flat. The epidermis has to grow large like this so that it can cover the new, larger body of the animal once it sheds its old exoskeleton. Now, once the epidermis has reached its final size, it starts to secrete a new layer of cuticle, right? That's the new cuticle growth. Now, the cuticle has to reach a sufficient thickness before the old cuticle can be shed and the new cuticle can be exposed to the outside world. If the new cuticle is too thin, it won't be strong enough to support the animal's body. Okay? Now, meanwhile, the older layer of cuticle, the innermost layer of that old cuticle, it's getting thinner and thinner. You can see here, as all these stages pass, this inner layer gets thinner until you're just left with the outermost, hardest layer of cuticle. Okay? When the time is right, and this new cuticle layer has grown sufficiently thick, enzymes will be secreted into this space between the new cuticle and the old cuticle. Those activated enzymes are going to facilitate the breaking apart of the old cuticle to create a gap that the animal can leave through. So this happens in different locations on different animals, but in all cases, the result is the same. There's a break that develops somewhere in the body. The animal is able to move itself out of that break in the old exoskeleton, and it looks a lot like they're taking off an old set of clothing, like how we would pull off a sweater or a pair of pants, right? You don't just rip your clothing to shreds when you get out of it. You pull your limbs out of the clothing so that it doesn't tear, right? You exit your clothing through an obvious hole. The same thing happens with the old exoskeleton. Okay? When the animal has backed out of this hole in its old exoskeleton, that's the final stage of shedding. Okay? The animal then has to wait for this new layer of cuticle to dry and harden after it's being exposed to the open air. Once it's hardened, the animal can go about its business. All right, let's move on to our last class of skeleton, the endoskeleton. Remember that endoskeletons, we talked about this in lab, they are internal support structures, and even more importantly, they're living tissue. Exoskeletons were made of non-living material, non-living chitin in particular. Endoskeletons, though, are different. They have living cells incorporated into them, so they are a true type of tissue. They do protect the internal organ systems of vertebrates, but because they themselves are inside the body, hence the name endo, endoskeletons cannot protect external structures like the integument. Okay? So endoskeletons do not protect all the living tissue in the body. They do provide significant protection, though, for those internal organs, and they have a lot of other functions as well. As you would expect, they help to maintain the shape of the body, but what might be surprising is that endoskeletons also participate in things like blood production. Bone marrow in the vertebrates is the source of blood cells. This is especially true for mammals. It's a process called hematopoiesis. That's the process of new blood cell production in the bone marrow. Endoskeletons also serve as a reservoir or a storage system for minerals. Bones mainly store calcium and phosphate, and the body can actually dissolve the bones if it runs short of either of these minerals. Okay? So people who experience starvation oftentimes have brittle bones because their body has raided their bones for calcium and also phosphate in order to just survive. Next function, movement, just like you would expect. All skeletons help to facilitate movement. In the case of endoskeletons, the bones and the muscles are going to work together to generate and transfer forces, to move these bones around to help the entire body move. All right, let's talk about some endoskeletal structures, starting with the histology of the skeleton. Right? Once again, this is the tissue level of organization for these organs. The organs of the skeletal structure are the bones themselves. The bones are made up of tissues that consist of living cells surrounded by hardened, non-living osseous tissue. This osseous tissue is composed mainly of calcium phosphate and calcium carbonate. This is the matrix. This osseous material is the matrix, which should remind you that bones are a type of connective tissue. Now, this tissue has quite a few specialized structures you need to know about. The first are the living cells themselves, plus the chambers in which they reside. Osteocytes are literally bone, hence osteo, and cell, hence site. These bone cells regulate bone mass and bone regrowth. If the bone is broken down or if the bone is literally broken, these are the cells that are going to repair or replace that missing material. And these cells need space in which to live. Remember, all of this matrix here that's filling in the space of the bone is solid calcium carbonate and calcium phosphate. The holes that you see here, these dark spots, these are little caves, little openings called lacuna. These little openings are where the living cells reside. Okay? These hollow spaces are where the cells themselves can be found. Now, these cells have to be able to talk to each other to coordinate things like growth. They do that using chemical signals that are passed through these little black squiggly lines here. These are called canaliculi. They're actual passages, tunnels, that connect the various lacuna to each other so the osteocytes can communicate. Now, living cells are going to need support. They will need nutrients. They will need an oxygen delivery system. They'll need a way to get rid of waste and CO2, which means they're going to need access to a blood supply via blood vessels. Central canals are larger openings, larger tunnels in the bone where you can find blood vessels and also nerves. Finally, you have these faint circles of material you can see here surrounding the central canals. These circles are what make bone look a lot like a cross-section of a tree trunk. They almost look like tree rings. These are called lamellae. Okay? They are circular layers of hardened tissue. They make up the matrix. This is where that calcium carbonate and calcium phosphate is found. Now that's the microscopic anatomy of the bone. Now let's take a look at the macroscopic anatomy, large-scale anatomy. So when you look at any given bone in the body, this is a femur, it's just an example, here's what you're going to see. The bone is going to be covered in a hardened outer layer, which is made of what's called compact bone. It's called compact because it's really, really solid. It has virtually no living cells in it, or it has a few, but not many. This is meant to be the hard, supportive outer shell of the bone. Now the ends of the bone are a different story. This is where you're going to find looser bone material. Okay? It's spongy. It has a lot of openings and holes in it, just like a dish sponge would. This is the location where you're going to find that bone marrow we were talking about, which is the source of our red and white blood cells. This is where those blood cells are being produced. Red bone marrow is what you find at the ends of the bones, and it's red because of those red blood cells that are being produced. Okay? So at the ends of the bones, you find red marrow, but what do you find if you look in the center of a bone? Well, that central cavity is actually hollow, and it's filled with a different kind of marrow called yellow bone marrow. Okay? Yellow marrow is fatty. That's why it has that yellow color. Lipids often have a yellowish kind of tinge to them. Okay? This yellow bone marrow also contributes a little bit to blood cell production, but it's primarily responsible for making new bone cells and storing energy in that fat. Now, associate structures with the bones are just as important as the bones themselves. Cartilage is a connective tissue that holds the bones together and also creates buffers or pads between them so the bones don't grind against their neighbors. Cartilage is another one of our connective tissues. Remember, it's composed primarily of collagenous fibers, mostly collagen, right? And also a set of specialized cells called chondrocytes, literally cartilage cell. Class chondrichthyes, our sharks, skates, and rays, are famous in part for having skeletons made entirely out of cartilage. But interestingly, all embryonic vertebrates actually start out this way. Every embryonic vertebrate, including those that later grow on to grow an entire bony skeleton, start out with a cartilage-based skeleton in utero when they're developing. That cartilage is then replaced by bone in animals like the amphibians, the reptiles, birds and mammals, and the osteichthyes, the bony fish. Now, cartilage can grow adaptively with the rest of the body, and it can be repaired if damaged, but it's slow going. And the reason is there's not a lot of blood supply in cartilage. There aren't very many blood vessels. And the less access you have to the resources carried by blood, the slower the work is you're going to be able to do if you're having to repair damage, because all of that rebuilding energy and rebuilding material is carried in the blood. Now, where cartilage and bones most famously come together are at structures called joints. Joints are the junctions between bones that help facilitate movement, right? So things like your knee joint, pictured here, your elbow, those are classic examples. There are 4 types of joint that we are going to be concerned with. There are a lot more than just these 4, but these are the 4 most common across all animals, not just within humans, but across all animals. Now, before we get to those, let's look at the general sort of generic structure of a joint. Joints are, like we said, where bones come together. Bones are held together at joints using connective tissue called ligaments. Ligaments are very, very strong. They're fibrous. They are not stretchy. There's a lot of collagen here. This is what holds the bones together in the right orientation at a joint, so they move against each other properly. Now, there's a lot of risk of damage when you put 2 bones next to each other and allow them to move. The bones themselves are hard, right? Calcium carbonate is a lot like a rock. You don't want the bones grinding against each other because it's like grinding 2 rocks together. There's going to be damage done. So instead, the bones are separated from each other by a series of structures that rely on cushioning and lubrication to keep the movement of the joint smooth. Synovial fluid is a fluid secreted by synovial tissue, specialized type of tissue that's found underneath ligaments. This synovial fluid acts as a lubricant. Okay? It helps to smooth the movement of the bones against each other. Okay? Now, synovial fluid isn't found in isolation. It's contained within this sac. The boundary of this sac is a membrane called the synovial membrane. This thing kind of looks like a pancake-shaped water balloon. It's almost like having a water mattress or a water bed stuck between these 2 bones. But instead of water, you have this synovial fluid that is filling this open space inside the balloon. Okay? This creates not only lubrication from the synovial fluid, but also cushioning because of how much fluid there is pushing the bones apart. But in addition to that, you also have pads of cartilage found on either bone. In this case, it's the hip and then the head of the femur, the leg bone. Okay? Each of these bones is capped by a layer of cartilage that also provides additional cushioning to keep these bones from actually touching each other. Now let's talk about these different types of joints. Here are your 4 examples that you need to know about. The first are called ball and socket joints. A socket is a round indentation. So if you look over here, in this case, this is the socket. It's a structure called the acetabulum. It's found on the hip bone. This is where the head of the femur goes, the head of your thigh bone. Fits right into this round indentation. Okay? Here's a simplified diagram of it. Imagine this cup is hollowed out so that this ball fits perfectly inside of it. This kind of joint arrangement allows for rotational movement. It means you can move that joint along several planes. The shoulder and the hip are the great examples of this. You can move your shoulders around in different positions. You can swing your arms around in a circle. You can move them forward, backwards, up and down. There's a lot of range of motion using a ball and socket joint. Now compare that to a hinge joint. Hinge joints allow for movement along a single plane. Think of this as being like a door, right? The hinges on a door allow the door to swing open and shut. And that's it, right? It can't lift up and swing up and hit the ceiling. It can't fall down and hit the floor. It can swing out or back in, and that's it. The knee joint works in the same way. Okay? This is a great example of a hinge joint. You have sort of an indentation here that's elongated, fits a rounded cylinder on the opposite bone, and it allows for this single plane motion, right? The true elbow joint is another example of a hinge joint. Think about how you can bend your elbow flat or bend it up so that your arm touches or your hand touches your shoulder. But that's it. You've got this 180 degrees of motion and nothing else. Now, near your actual elbow, which is what's being depicted here, you actually have another joint. It's where your lower arm bones come together, right? Your two bones in your lower arm are the radius and the ulna, okay? They are actually connected to each other using a joint called a pivot joint. Pivot joints allow for twisting rotation. Works kind of like a peg in a hole, okay? So this peg can spin around, but it can't bend outwards, right? If you think about how your lower arm bones move against each other, they always stay next to each other, right? But if you hold your arm out straight in front of you with your palm facing the ceiling, without moving your elbow at all, hold your elbow steady, you can turn your palm so it faces the floor. And what happens when you do that is these two bones in your lower arm, they cross over each other, right? That's allowed to happen because the radius can rotate in this little sheath of ligament that attaches it to the ulna, okay? So the radius can spin in this ligament, and that's what allows you to turn your palm face up or face down without turning your entire arm. There's another pivot joint that connects your skull to the top of your vertebra. That allows you to rotate your head side to side. And then finally, we have gliding joints. Gliding joints are where flat bones connect to each other. And they don't lift away from each other. They stay touching, but they can slide against each other in all of these different directions. You see this kind of joint where you have clusters of lots of small bones that are all on top of each other? Think all of the bones in your foot and all of the bones in your wrist, right? Your wrist bones, when you rotate your wrist, just glide across the surface of each other. Your vertebrae work the same way. When you bend your back forward or backwards, side to side, your vertebrae slide, but they don't lift away from each other. Those are all gliding joints. Now, the skeleton overall can be subdivided into two regions. The axial skeleton makes up the axis of the body. This is the skull, the vertebra, and the ribs, right? Central axis of the body. Everything that attaches to the axial skeleton, meaning the hips, the legs, the shoulders, the arms and the shoulder blades, all of that makes up the appendicular skeleton. Think appendages, arms and legs. Okay? This includes two important structures, the pectoral and pelvic girdles. A girdle is a circular supportive structure. The pectoral girdle is found up here by your pecs, your pectoral muscles. It's made up of your clavicle and your shoulder blades. This provides support for your arms and where they attach to your body. The pelvic girdle is made up of your hip bones, okay, and the base of your vertebral column, your sacrum, okay. This is also circular in shape. It provides support for your legs. Now there's one last aspect of the skeletal system that we need to go over. This has to do with how we classify animals based on their cranial structures. The cranium is this part of the skull. It's the part that actually houses and protects the brain. Okay? Now, there's three different types of craniums that we're going to talk about: anapsid, diapsid, and synapsid. We use these three cranial classifications to differentiate between different groups of animals. Now, the way that you wind up classified into one of these three groups is based on the presence and the number of holes that are called temporal fenestra. These are openings in the skull posterior to the eye orbit. Posterior means behind. Okay? In the case of these animals, this is a sea turtle skull. Okay? This is just a generic skull showing you what a skull looks like that has no temporal fenestra. Okay? When these holes are present, they look roughly like this. Okay? This is the eye socket. Here's the nostril. These are postorbital fenestra. Okay? Now, when they're present, they're So when they're present, they're very obvious. When they're not present, it's also obvious the skull is very smooth. Now, what is their function? There's still some debate about this. We're not 100% sure, but it appears that these openings in the skull evolved to create more room for jaw muscles. All right? So if you want to have a really powerful bite, you're going to have to attach a whole lot of muscles to your lower jaw. So when it pulls shut against your upper jaw, it closes strongly. Right? So if you want your bite to be powerful, when your lower jaw closes, it's going to have to close fast and hard. That's going to require a whole lot of muscles and places for those muscles to attach. It's possible that these openings in the skull created more space for those muscles to grow. Now, you can have either no temporal fenestra as a vertebrate, you can have one of them, or you can have two. The animals that lack temporal fenestra entirely are called anapsids. Apsid means arch. These things kind of look like archways in the skull, little arches made of bone. An means without an arch. Okay? The anapsids are represented by the chelonians. These are all of our turtles and tortoises. Right? Turtles and tortoises. Turtles and tortoises are the only reptiles that are anapsids. The rest of the reptiles are diapsids, di meaning two. These guys have two ancestral temporal fenestra, meaning some of the more modern species have lost them, but ancestrally, all other reptiles had two. They're positioned posteriorly above and below the eye orbit. This is the orbit. Okay? Posterior means behind. Here's one above. Here's one below. Okay? You see this cranial state in the lepidosaurs, which is the clade that includes lizards and snakes, and also our old friends, the archosaurs, which include the dinosaurs, the crocodilians, and the birds. You can see the lepidosaurs down here. Okay, so here are the diapsids. Follow this line and you get to the lepidosaurs. Okay? Follow this line and you get to the archosaurs. Now take a look at these images. It can make it a little easier to understand what we're talking about. Okay? When you look at, say, a crocodile skull from above, you'll see there are two eye sockets, and then behind them, these two holes in the top of their head. These are the temporal fenestra that are above the eye. To either side of the eye are the temporal fenestra that are below the eye. It's easier to see on this Tuatara skull. Okay, this is another very ancient lineage of reptile. Here's the eye orbit. Okay. Here's the upper temporal fenestra. Here's the lower one. And here's the arch of bone that separates them. Remember, diapsid, two arches. Here is the arch created by these two holes. There's one on the other side of the skull as well. You see this even in the dinosaurs, right? Here's the eye socket. And again, here is the lower temporal fenestra. Here's the upper one. This big hole here, this is part of the sinus cavity. These guys had an amazing sense of smell. Our last group are the synapsids. The mammals are the only extant group of synapsids that we have. Okay? Synapsids have only one temporal fenestra. It's positioned behind and below the orbit. Okay? So it's down here. Now, in modern animals, the sphenoid bone, which is up in here, it's expanded to cover the opening, leaving only the zygomatic arch. Right? Let me show you what that looks like. Zygomatic arch is your cheekbone. Okay? So this is an ancestral mammal called Dimetrodon. Okay? Dimetrodon had an eye orbit here, like you'd expect, and a very obvious temporal fenestra behind it. But in modern day mammals, this temporal fenestra is hard to see from the side. Okay? So here's a slightly awkward picture of a human skull. If you were to look at the human skull from directly to the side, so you could see the eye socket easily, you'd see the cheekbone here, and just behind the cheekbone is this hole. This hole goes all the way through. Okay, so this is back close to where your ear is. Here's your ear hole. Okay, so just up and in front of your ear is the zygomatic process, and above that, you can't feel it, but there's a hole there, a gap. This is where your muscles for your jaw go. Okay, they go right through this hole down to your lower jaw. Now, if you flip a mammal skull over and look at it from below, like this coyote skull, suddenly these holes become very easy to see. They are massive openings here and here. Right? So we're looking at this coyote skull from below. And again, this is where our jaw muscles connect from our temple down to our mandible. All right, that's it for this lecture. If you have any questions, please let me know. Otherwise, I hope you enjoyed. Have a great night.